Hi, I'm Joanna Bauman at Heritage High School, and today we're going to talk about how to do a plan diagram properly, for the British way. So, you have in front of you what they define in paper 32, made June of 2011, as a slide of a stained transverse section through a stem. The first requirement is to draw a large plan diagram of the whole of the transverse section, not a portion, a whole. Label the epidermis and the xylem. So you're going to begin by starting with your outer edge. Remember that when you draw a plan diagram, you need to make sure that you don't leave any little wispy tails. So you want to have everything going in one smooth motion. Unfortunately, my whiteboard isn't large enough to get the entire picture in there, and I can't shrink it down. So we're going to just have to sit here and do what we can on the whiteboard. I'm not going up above it. Ready? I'm going to start at the outer edge. Don't pick up your pencil. Okay? You can continue on the other side. I'm going to go ahead and start up here with the epidermis inner layer. Notice that your epidermis layer here is one cell layer thick. Okay? Continue on the other side. It's hard to see where I'm going because of the video. Back along to the inner lining of the epidermis. It doesn't have to match right up, but when you're drawing, you want to be pretty consistent in the width of your particular layer. Okay? So there we have the outer epidermis. If you're going to label this, because it does say to label the epidermis and the xylem, you want to put your epidermis written outside of it. You draw your line horizontal and stop at the edge of the epidermis. Now we're going to go and deal with the inner structures. First of all, you can see here that we have this darkly stained tissue that's going to be the xylem. The reason it's darkly stained is because it contains high amounts of lignin. All right, and so we are going to, is it lignin? Yes. Is it lignin? Pause.
If I pause it, I can't start it back up in the same spot.
It's keep going. It is. Xylem's lignin.
Okay, that's what I thought. Okay, well let's go back because I'm having a moment here. Let's try again. Start at your edge and go all the way around. Get your tissue. Keep that line going as smooth as possible. Make sure your end nicely overlaps. You can see that there's two distinct layers of tissue in here. So you want to put in a line to designate the difference between those two tissues. And now you're separating that vascular tissue from your pith. Make sure your tails don't show up. Try to make them nice and smooth. Cambridge will grade you down if you don't. Okay? Now, they specifically said to label the xylem. They are not differentiating between the two layers as to which one is xylem and which one is phloem. So in this case, you would just want to draw a line horizontal until you reach the first layer of vascular tissue. That right there satisfies the requirements for this particular question. However, there are additional questions where you're supposed to calculate the diameter of the total diameter of the stem to the diameter of the central pith. In order for you to accomplish this task, you would have to actually know what is the pith. And so this in the center is the pith. Okay? In case you're not familiar then, this outside is the cortex. You would not, under any circumstances, want to actually write those labels in there on your plan diagram. We're merely doing this for your own educational edification. So you can calculate the ratio of the total diameter of the stem to the diameter of the central pith. So you're going to be wanting to measure this to that. You would want to sit and measure both of them using your gradical, because you're looking at a slide. And so you look and you calculate the number of eyepiece gradical units that are across on this line that goes across the total diameter, and then the number of eyepiece gradical units that you would have going across the pith. And then you would do the ratio. When you do a ratio, you want to leave it either as a fraction, or you want to, but you always want to have the largest number first in this particular instance. Okay. Now, the next question is, state one observable feature of the epidermis that supports the conclusion that this stem is a plant growing in a dry habitat, which means that this plant is a xerophyte. Okay. This plant has adaptations that are going to allow it to deal with lack of water in its environment. Hold this one out. Okay. So, one observable feature. And that observable feature is right here. The little indentations in this particular stem mean that the water that might escape through the stomata in that particular part of the epidermis, the little water molecules are actually going to remain right here. And because wind movement is out here, those water molecules are not going to be carried away as quickly as, say, a water molecule that might transpire from a stomata out here. These will be carried away. That will create a greater water potential gradient than in here in these little crevices. And so those crevices enable the plant to keep from losing its water as easily. And so that gives you your feature and your explanation for how it functions, how it reduces water loss. And then they want you to make a large drawing of 3 adjacent, that means touching, cells from the central pith, and they want you to label the cell wall. So if you were going to draw these particular cells, and I'm going to draw them off to the side so that you can see this, the cells that I am going to select, 3 adjacent cells from the pith, so if you're looking at this on a microscope and you wanted to draw those cells, you would now sit and draw those cells. And you always want to make sure you use the space provided. So it's not going to be small, it's actually going to be large. Hang on 1 second. I want to make sure it actually looks like what you're doing. There. Now they want you to sit there and label the cell wall. Can't get to my test question. And that completes question number 3 on that paper.